When blood bank specimens arrive in the lab, they need to be processed immediately. They usually need to be tested and resulted in a very rapid and efficient manner. Therefore, you need a very efficient and organized tech doing the testing. Messy Malice seems to struggle with this. She's not prepared, she is not dressed as she should be, she's not familiar with her equipment, and she struggles to make sure that she has everything that she needs. Shoot. Being unorganized will definitely delay the test results. Miss Messy was not going to work out in the blood bank. Instead, we've replaced her with Efficient Elise. She's going to make sure that she has all of her equipment out and ready to use when the specimens arrive. She's going to make sure that everything she places is within reach. She checks to make sure that the reagents are in date and they are exactly what she needs. She makes sure that her racks are set up and her tubes and her pipettes are as she needs them. So she'll take out her agglutination viewer and put it where she'll be able to reach it and read her tubes easily without going into other areas, other people's areas. Often in the blood bank, you're going to have your workstation set up before you get there. It'll just maintain setup. Notice when she's labeling tubes here, she has the tubes laying down on the ground. Don't try picking them up and labeling them. It takes twice as long. If you label them down, it's just like writing on a piece of paper. And notice she labels them at the open end area. If you label them close to the bottom, it makes it hard to read your agglutination. So label the tubes up at the top where you'll be able to read your labels, but it won't interfere with reading. To pick up the tubes, the easiest way to do it is just to press on the top, and you can pick up multiple tubes at a time, and then place them in your rack. It's most efficient to take your tubes out, label them, and then put them in your rack. Don't try to take your tubes, put them in your rack, and then take them out one at a time to label. Lay the tubes down and label as you need them. If you have 3 different patients, 3 different specimens, make sure you keep them separate. Don't mix them together. Keep all the tubes separate for each patient. Notice how she's labeling the tubes separately for each patient, and she'll make sure that those tubes only stay with that patient. It's a very easy way to mix it up if you start having your tubes mixed up. Notice how she checked the label on that specimen tube, and she's labeling those tubes specifically for that particular specimen. And then she'll put each of those tubes in line with that specimen. If there's a 3rd specimen, she can either get another rack, or she can make sure to have it in a different area of the same rack, but never have your specimens mixed together. When you're doing a plasma aliquot, you want to have the 4 identifiers on your aliquot, meaning that specimen will be labeled with all the proper identification the original specimen had. And make sure that you initial that aliquot so someone who is following you knows who separated that plasma. So the plasma has now been separated, making it so it'll be easier to get that specimen tested later if for some reason we needed to cross-match additional units, etc. What's remaining in the tube would be packed cells. So here we see the RBC packed cells. This is cells that have been centrifuged down, and all the red blood cells are on the bottom. The main reason for packing these cells would be to standardize the crit. If you get a patient down who has a crit of 20, and you get another patient down who has a crit of 40, they're going to have very different concentrations of red cells, and making a 3 to 5% suspension is more tricky. But if you pack the cells, centrifuge the cells, it's a standard process because all the cells will be down at the bottom. It doesn't matter if the person has a 20 crit or a 40 crit. All the cells will be down at the bottom, and you're going to take 1 drop of those packed cells in a pipette and put it into a transfer tube there. Once you put it in, you're going to fill that tube halfway with saline. Now the next step will be to make a 3 to 5% cell suspension. So to do that, she's going to label another tube. This tube will be discarded at the end of this testing period. So it doesn't necessarily need those 4 identifiers on it, but it does need to be labeled to where the tech making this aliquot can identify it. In a 10 by 75, to make a 3 to 5, what you're going to do is label the tube and then fill that tube half full with saline. Once it's half full with saline, place it down, and you're going to pick up your pack cells, and you are going to check the label. Always check the label. Every time you pick up a tube, every time you touch a reagent, you're going to check the label to make sure you have in your hand what you're expecting. Then take a pipette, transfer pipette, and put it into the pack cells and pick up enough to get at least 1 drop in there. So always there's going to be extra, which is fine. So put in 1 drop into your tube with the saline, squirt the excess back into the tube, and then using the same pipette, mix it in the saline. Once it's thoroughly mixed, you should have a 3 to 5% suspension. You'll be able to compare this to your reagents. We don't actually have the cellular reagents in the lab at this moment, but you can compare it to your reagents to see whether or not you have a 3 to 5% suspension. Okay, now you're ready to do testing. So at this point, you can use the 3 to 5% suspension for testing your antigens, and you're going to use the plasma for testing your antibody response. At this point, we can continue on with testing to adding our reagents in the specimen to all the tubes.